Hello everyone, welcome to the channel and today we will discuss about hemoglobin. So, hemoglobin is the respiratory pigment of RBCs. So, the red color of RBCs is due to hemoglobin. Hemoglobin basically contains a protein which is called globin and which has 2 alpha chains and 2 beta chains in case of adults and 2 alpha chains and 2 gamma chains in case of fetus. So, and heme molecule which is basically made up of iron. So, in case of hemoglobin, the globin is conjugated with heme. Heme molecule contains 4 pyrrole rings with iron in the center. Average hemoglobin content is 14 to 16 gram per deciliter. At birth, it is 25 gram per deciliter, and after 3 months, it is 20 gram per deciliter. After 1 year, it is 17 gram per deciliter. From puberty onwards, it is 14 to 16 gram per deciliter. In adult males, it is 15 gram per deciliter. And in adult females, basically the females of the reproductive age, it is 13 to 14 gram per deciliter. It is less because of menstruation, and after menopause the level is almost 14 or 15 gram per deciliter. Now, functions of hemoglobin, the 2 basic functions are transportation of respiratory gases such as oxygen, transportation of oxygen and carbon dioxide, and it acts as buffer and maintain acid base balance. It is also source of bilirubin. Now, hemolysis is a condition where there is escape of hemoglobin from the RBC in the blood. This is caused by hypotonic condition. Hypotonic conditions such as if we place the RBC in a hypotonic solution due to certain drugs and toxins. Now, there are some types of hemoglobin. Normally, the hemoglobin is of 2 types. Normally, the hemoglobin is of 2 types. Hemoglobin 2 types. It is normal hemoglobin. One is adult hemoglobin, which is also known as HbA, and one is fetal hemoglobin, which is known as HbF. Now, abnormal hemoglobin, it is of 2 categories. Abnormal hemoglobin is there, and the reasons are due to hemoglobinopathies and hemoglobin in some disorders such as thalassemia, which is a blood disorder. Now, hemoglobinopathies occurs because of genetic disorder caused by abnormal polypeptide chains of hemoglobin, and based on this, hemoglobin is of the following types: hemoglobin S. So here it is found in sickle cell anemia, where the RBCs become sickle shaped. In this type, the alpha chains are normal in globin and beta chains are abnormal. Next is hemoglobin C. In this, the beta chains are abnormal. It is found in people with hemoglobin C disorders, which is characterized by mild hemolytic anemia and splenomegaly. Splenomegaly means enlargement of spleen, and hemolytic anemia means anemia due to destruction of the RBCs. Next is hemoglobin E, which is here the beta chains are abnormal. It is present in people with hemoglobin E disease, which is characterized by mild hemolytic anemia and splenomegaly. Now, hemoglobin N, it is abnormal hemoglobin present in the form of hemoglobin M. It is mild hemoglobin present in the form of methemoglobin. It occurs due to mutation of the genes of both the alpha and beta chains of globin, resulting in abnormal replacement of amino acids. It is present in babies affected by hemoglobin M disease, or which is also known as blue baby syndrome. It is an inherited disease characterized by methemoglobinemia. Next is hemoglobin thalassemia and related disorders. The polypeptide chains are decreased, absent or abnormal. In alpha thalassemia, the alpha chains are decreased, absent or abnormal, and beta thalassemia, the beta chains are decreased, absent or abnormal. Some abnormal hemoglobin in thalassemia are GHI, BARTs, etc. Now, there are certain abnormal hemoglobin derivatives are found by carbon monoxide poisoning due to some drugs like nitrates. It is caused or formed by carbon monoxide poisoning or due to some drugs such as nitrates and sulfonamides. One is carboxyhemoglobin, abnormal hemoglobin, carboxyhemoglobin. It is found in abnormal hemoglobin derivative found in when iron molecule of hemoglobin is oxidized. It is normally 1 to 3% of hemoglobin in the form of carboxyhemoglobin. Carboxyhemoglobin is the abnormal hemoglobin derivative formed by the combination of carbon monoxide with hemoglobin. All of us know that carbon monoxide has much affinity for hemoglobin than oxygen, so it easily combines with hemoglobin. Next is methemoglobin. It is an abnormal hemoglobin derivative formed when iron molecule of hemoglobin is oxidized from natural ferrous state to ferric state. In normal condition, the iron molecule is in ferrous state, but in case of methemoglobin, the iron molecule is in ferric state. Normal level is 0.6 to 2.5% of total hemoglobin. And methemoglobinemia is a disorder with high level of methemoglobin in blood. Factors include daily life factors, which are common factors, industrial chemical exposure, certain drugs, hereditary trait, etc. Next is sulfhemoglobin. Sulfhemoglobin is the abnormal hemoglobin derivative formed by combination of hemoglobin with hydrogen sulfide. Sulfhemoglobin with hydrogen sulfide. Hydrogen sulfide, it is hydrogen sulfide H2S. So, normally sulfhemoglobin is present in blood in very negligible amount, which is less than 1%, but when the level rises above 10 gram per deciliter, it is known as cyanosis. So, these are about normal hemoglobin its functions, types of hemoglobin, both normal and abnormal, and some abnormal derivatives of hemoglobin. So here I am ending my today's discussion. See you in my next video, and if you guys like my videos, please subscribe the channel. Thanks for watching.